Thank you for joining us for this exciting session discussing the future of drug discovery. The title may sound ambitious, but discussing ways to improve drug discovery and development has always been a core focus for the ON Helix conference. And this session is no exception, with a review of 3 really innovative technologies, followed by a discussion on their transferability and added value to existing drug discovery processes. And so to share that session, who best placed than someone with a strong experience of progressing candidates to clinical ready stages to ask the right questions to our presenters and panelists. So I'd like to invite and thank Suzanne Dilly, the CEO of ValiRx, who have accepted our invitation. And so Suzanne, over to you.
Thank you, Aileen, and thank you everyone. Welcome to the ON Helix Future of Drug Discovery panel. Joining us today, we have 3 leaders of exciting technologies and exciting companies. And just to introduce these to start with, this is Ajay Mistry, founder and CEO of Oppilotech. We have Kam Dhaliwal, Senior Vice President of Business Development and Licensing of bit.bio, among other roles. And we have Martin Bittner, CEO of Arctoris. Now, the format of this session today is that each is going to give a presentation of their technologies and their companies. And then we're going to open up for a more general conversation. So at that stage, please, everyone, feel free to put your questions into the chat box. And we look forward to discussing exactly what everyone is up to and where we see the future of drug discovery going. So over to you, Ajay.
Thanks, Susie. So as you know, I'm the CEO and founder of a company called Oppilotech. What we do is we build computational models of cells, and then we use the model to find points of intervention, so drug targets. Then we develop drugs against those targets. So we're building a pipeline of early stage drug discovery programs in the oncology and the infective diseases area. So can I have my first slide, please? This is an image of one of the most studied organisms in the world. It's an image of the bacterium Escherichia coli. And it was first discovered by a German pediatrician called Theodor Escherichia in 1885. And he published a paper in 1885. And since then, there have been literally hundreds of thousands of publications on this particular bacterium. And there have been various Nobel Prizes as well, based on the biology of the E. coli bacteria. So I think we can safe to say that it's you know, this particular organism is well studied, and we seem to know a lot about it. Next slide, please. So if we look at E. coli in numbers, it has something like 4,225 genes, over 10.7 million different RNA species, over 132 million different proteins, and over 300 million different metabolites. So I think it seems to tell a different story in terms of us understanding the biology of E. coli. Can I have the next slide, please? So if you look at these components I just described, what's amazing is that these all fit into a cell which is 3 micrometers by 5 micrometers in a volume of 0.7 micrometers cubed. So just think about that all those components fit into a cell. And in addition, the bacteria is not like a balloon with these components randomly swirling around. It's structurally very well organized. So this is an image from a famous structural biologist called David Goodsell, and you can see it's part of an E. coli cell sliced in half. And you can see that the proteins and components are very well structured. So what I'm trying to say is that although we've been studying this E. coli for over 137 years, there is still a lot of unknowns, still a lot of biology that needs to be discovered. And what we've tended to do in the past is take a reductionist approach. So we've looked at one pathway at a time. What we need to do to get a better understanding of the biology is to take a more holistic, integrated approach that links these different components together. And that's what Oppilotech does. So we've developed a platform to computationally model cells and cellular processes, and we look at biology from a more integrated perspective. We then use this knowledge to find points of intervention, drug targets, and then develop drugs against those targets. Can I have the next slide, please? So what are the input parameters for our computational model? So some of the input parameters that go into our model are things like gene transcription and translation rates, catalytic rates of enzymes, messenger RNA half-life, regulatory mechanisms, and enzyme concentration. So those are some of the input parameters. And what comes out of our model? Things like, I guess we are looking for viable drug targets so we can develop drugs against those particular targets. The sensitivity of these targets, whether there are any bypass mechanisms, and any new regulatory mechanisms. So those are some of the outputs of our model. Can I have the next slide, please? So E. coli belongs to the group of bacteria known as gram-negative bacteria. As you probably, a lot of you know, the gram-negative bacteria have this extra outer envelope, which is made up of LPS, phospholipids, and peptidoglycan. So we initially focused our modeling on modeling this biosynthesis of the outer envelope. And I'll show you an example of our modeling in action. Can we have the next slide, please? This was an animation representing an E. coli cell. So the dots were representing enzymes, substrates, and metabolites involved in making a component of the outer envelope, the LPS. And what we could do is push down on different parts of that network and see what impact it has on the biosynthesis of these different components. Can I have the next slide? So here, what we were trying to illustrate was by pushing down on different parts of the network, you could see that the lipid A, in this case, component was going down. And basically, to cut a long story short, what we did was we identified a enzyme involved in the biosynthesis of lipid A, and we used that as a drug target. So next slide, please. So yeah, so we used our computational model of E. coli to find a drug target, and then we developed drugs against this particular target. And to cut a long story short, what we found is that after just one round of a hit to lead campaign, we've got antibacterial activities. So we've got a program now, which is a first-in-class program targeting lipid A biosynthesis, and it's an antibacterial which we want to take towards the clinic. So we've gone from computational model of a cell, developed, identified a target, developed small molecules against the target, and taking that into the, and we're taking that program towards the clinic. Okay, can we have the next slide, please? So what we did, so that was the modeling around the around the bacteria. So what we next did was look to modeling mammalian pathways. Of course, mammalian pathways are much more complex. So we hooked up with Simon Cook from the Babraham Institute, who's the head of signaling there. And we worked together with Sam to model the MAP kinase pathway. Today, this model contains at least over 180 nodes. And what we've done is we've managed to identify an interesting target. And we've just started to develop chemical matter against this target. And we're going down the PROTAC route for this target. Next slide, please. Another program we're doing in the mammalian world is in modeling the DNA damage response pathway. So, we're working with a group at the University of Sussex. So that's the Francis Pearl and Lawrence Pearl's group, who are experts in this area. And we're looking to model aspects of the DNA damage response pathway, especially those implicated in synthetic lethality. Again, the idea here is to find viable drug targets and develop drugs against those targets. Next slide, please. So to summarize, we are just scratching the surface when it comes to understanding biology. I hope the explanation around the E. coli you were able to appreciate that from that. And we're shifting away from reductionist approaches, and we need to take more of a whole cell, holistic perspective when it comes to understanding biology. And to do this, we've been developing a computational model to help us understand and discover new biology. And we use the models to identify viable drug targets. And we're building our own pipeline of drugs in the anti-infective and oncology space. Can I have the next slide, please? So I'm going to leave you with this particular slide. So as you probably know, last week, Donald Rumsfeld, the former Secretary of Defense, died. And his infamous quote was all over social media. And I'll just read it out to you. There are known knowns. There are things that we know we know. We also know there are known unknowns. This is to say, we know there are some things we do not know. But there are also unknown unknowns, the ones we don't know we don't know. So this was obviously said in the context of the Iraq War and finding weapons of mass destruction. But I think we can apply this this statement, especially unknown unknowns, in the world of biology and drug discovery. It's the unknown unknowns that can destroy a multi-billion-dollar drug discovery program. And what's interesting, and this is I found out last week, was Rumsfeld was very active in the pharma industry. He was the chairman of Gilead for quite a few years, and he was also a CEO of a pharma company called GD Cell, which was sold to Pfizer. And he was also on the board of Amylin. So just to end, what I'm trying to say is that with our computational modeling, what we're trying to do is figure out those unknown unknowns and hopefully develop better, more successful drug discovery programs.
That's great. Thank you, Ajay. I think that's certainly a good example of how Oppilotech will be able to shine a light on some of those unknowns. So moving on, Kam, would you like to introduce yourself and provide your presentation?
Yes, hi, thank you. Thank you there, Suzanne and Ajay. Thanks very much. I'm really pleased that I'm being able to attend the conference, and obviously looking forward to the discussion after this. So I'm just going to share my screen. Hopefully everything works, and I'm hoping that everyone can see my screen. I'm hoping that's approval. So I'll just move on. So the talk actually is really centered upon challenging biology. Can we code for human cells that will impact health? Now, many of you will have known all about IPSCs, and I mean, I'm of the decade. I remember running my first biochemical screens back in the early 90s, and I lived through the sort of industrialization of drug discovery, and obviously more recently with my adventures at Horizon Discovery around the CRISPR revolution as well. So I think looking at IPSCs and how we can actually code and systematically code for different iPSC-derived cell types and their impact, not only in basic research and drug discovery, but also from a therapeutic point of view. So this is really the journey I'm going to take you on, and what bit.bio are actually doing. So I think we all appreciate that medicine is at a crossroads. I think we can all understand that we've all probably read enough or been in the industry long enough to realize that there's a particular measure, a ratio that we measure, especially in pharma, around R&D spend and the number of biological license BLAs or new new chemical matter entities. And I think a number of articles will show you that our productivity compared to previous decades has really never been lower. And we live in a third age of how we are developing therapies. Obviously, in the early part of the 19th century, we're looking at small molecules. Then in the generation of biologics and targeted drug discovery. And now we live in an age where really with cell therapies hold a lot of promise to deliver effective therapeutics, especially in oncology. But there is what we call a common shared problem. And that shared problem in drug discovery and that same problem in developing more affordable cell therapies is based around how can we access high quality human cells? Now, if we were able to do that, what impact would that have on our drug discovery programs, either starting with basic research, target discovery, using more relevant translatable disease models to human biology rather than animal models? And what are the barriers we're trying to overcome? And these barriers relate to precision, consistency, and scalability, but also functionality as well. And these are the things that we at bit.bio are leading the revolution on in terms of addressing some of these challenges. And I'm hoping these couple of slides will give you some context of what we're trying to achieve. So this is a very famous slide. It talks about development biology, really starting with a single cell, which then over a developmental period, you have a multicellular organism that relates to a newt. Now, imagine a situation where you start off with human pluripotent stem cells, and you're able to effectively code in any different cell type. And not only that, but also then build out disease models that are relevant using technologies like CRISPR. Now, that is at the heart of what we are doing at bit.bio. And so some of you will be well aware that IPSCs have been around for decades and will be aware of conventional directed differentiation, and where there is a use of a manipulation of the external environment in terms of growth factors and media conditions, which lead to situations where you have good protocols, but there's a lack of purity, a lack of scalability and consistency. And then the work that was done by Yamanaka, where they're able to demonstrate that a fibroblast could be converted into IPS or ES cell. And then there's the lateral reprogramming, or what we call transdifferentiation of a fibroblast to a neuron. Now, what we are talking about today is what we call cellular programming, and that's forward programming. That's the work that was done by Mark Cotter, our founder, and Zhang back in 2013, where we are effectively coding in using transcription factors and forward programming to the cell type of interest. So this is at the heart of what we try and do at bit.bio and have demonstrated. So when it comes to cellular reprogramming, I always look at transcription factors as master control switches, but really they're the computer code for every cell type. And if you can identify these transcription factors, then code them in directly and have a vehicle to introduce into an IPSC, you can then program them to a specific cell type of interest. So much of this work has been built on the shoulders of giants: Yamanaka, Professor John Gurdon, Thomas Graf, Harold Weintraub, Maris Wernig. So we saw a tremendous upsurge in the amount of publications around cellular reprogramming soon after the work that was done by Yamanaka. But to date, there are only around 30 transcription factor combinations described for around 30 different cell types. So what happens when you don't know the transcription factors. And again, that's the story behind bit.bio. So that's where we have developed a high throughput combinatorial method using machine learning algorithms to be able to identify and also characterize functionally and validate those transcription factors and develop cell types. So at bit.bio, the barriers that we're trying to address is really around cell identity. And the approach that we take is, once we can identify the TF code, or the transcription factor code, we base this, as I said, on a software approach, which at heart is a method which utilizes a combinatorial approach of identifying transcription factor combinations that can be controlled and that are master transcription factors that can help program to a specific cell phenotype. And a key pillar of our platform is a genetic switch called Optiox. So I'm going to talk about this in the next slide. So Optiox is really, as I said, a glorified genetic switch. And this provides us with this fine level of control of being able to introduce transcription factors in a scalable method to be able to produce batches of consistent cells, and not only for CNS cell types, for any cell type that we so choose, as long as we have that code. Now, this is a video that is a time-lapse video of an IPSC which has been converted in a matter of days to a skeletal myocyte, and it begins to contract within 8 days. But it started as a single IPSC. So what does that mean? Well, if we can do this, and if we can deliver cells that are consistent, scalable, functional, pure, that is going to be a valuable tool for basic research and drug discovery and tox. We're developing next generation tox models with the help of alliances with the likes of Charles Rivers and one leading pharma company. We're also will be announcing in the future, very, very near term, a cell therapy partnership with a top 3 pharma company. And you can see that the technology fundamentally has applications not only in drug discovery and cell therapy, but in the future in biomanufacturing. Now, although I'm not talking about this specifically, we have a sister company called Meatables who are applying the same technology that I am describing to you today, but in artificial meat. And so this kind of gives you a vision of what we're trying to achieve when it comes to scale. So they're talking about trillions of cells and the impact that that could have for cell therapies and making them more affordable. I think that really is a great pointer. So we know that this technology can scale. And so a little bit about how we do it. So as I said, we start with we have a drug discovery platform, or a discovery platform, that really teases out the right type of transcription factor combination. We start with a library of up to 50 transcription factors per cell type. We randomly distribute them into an IPSC. We use technologies like single-cell RNA-seq to tease out the combinations that give us a specific cell type. We do some functional validation and characterization. So we test in cell whether or not a specific transcription factor combination is going to give us a designed phenotype of interest. And once we have our priority list of maybe the top 4 or 5 transcription factors, we then start building out cell types using our Optiox cassette, where we can incorporate these transcription factors into 2 safe harbor locus. We can then effectively code that into an IPSC and then generate the cell types that we want. And so whether they be myocytes, whether they be cell types involved in neurodegenerative disorders, whether they be involved in metabolics, etc. So there's a lot of there's a lot of flexibility in terms of the way we can use this technology to generate the cell types that we want. So we can do this at scale, we can do this consistently, we have defined populations. In terms of, you saw from the video, but the methodologies, we're not using directed differentiation, we're using forward reprogramming. So what does that mean? That means we can do this at speed. That means that the protocols are easy, they can scale, and they're more cost effective. And so this will have a dramatic bearing on how researchers can access not only high quality iPSC derived cells, but also disease models as well. And the key things that we drive for is looking at things like consistency, making sure that there's batch-to-batch reproducibility. Why is that important? Well, not only from a cell therapy context, but also from high-throughput screening context. We look at things like purity, so making sure that there are defined populations. Yes, you might have, for example, in a glutamatergic cell types, you'll have two different populations. One might be 80% glutamatergic, but there is a defined cholinergic population as well, but they're 100% neuronal. And researchers need to know that, and they need to know that they're consistent. Then there's scale. As I mentioned, there is no limitation. Where once you've got the code and you're able to deliver that transcription factor combination code using the OptiOx cassette into an iPSC, we can scale this into, as I said, in our sister company, is producing the bovine equivalent of skeletal myocytes and adipocytes in terms of trillions of cells. And then functionality. So looking at not immature function, especially in terms of CNS cell types, but more mature function, which is critical. And then speed. You don't want protocols that cannot scale and that are difficult to use and that are costly in terms of additional reagents and media. You want things that can happen in a matter of days. And that is and actually cells that are functional as well. And then finally, I just wanted to kind of touch upon how these are being used in the real world. Our cells are now being used in a primary HDS setting. We have strategic partners like Charles Rivers who are using our cells, which especially our glutamatergic neurons, which are synchronously active within 20 days. And really, we're pushing the boundaries with partnerships with Professor Hagan-Bailey at the University of Oxford, where we're combining our glutamatergic neurons with astrocytes to create more complex disease models. So we're looking at co-culture systems which can be made at scale.
That's absolutely brilliant, and that's a really good insight in seeing how cells can be identified as well as programmed. So I'm looking forward to more of the questions to help explore us
absolutely fine.
A little bit further in the Q&A later in the session. But moving on
absolutely
to Martin.
Fine. No problem. I'll stop sharing.
Wonderful, thank you very much. So I think now it's kind of my turn, and I'm very glad to be here. And thank you very much again, kind of to Susie in particular for sharing this very interesting session. And in terms of brief background, so my name is Martin Bittner, and I'm the CEO of Arctoris. I'm one of the founders of Arctoris. And I'm just going to show kind of some very few slides, just to give a very brief background regarding how we see the industry, the drug discovery industry, shaping up right now, what we see as some of the key challenges, and how we think things are going to progress from here. So if we basically briefly bring up the slides, wonderful. So in the next slides, what we're going to see is kind of what we define as the core challenges within drug discovery. One is quality. Data quality in drug discovery is oftentimes poor. And this is something which ties directly back into what Kam has just been talking about, which is that we know there is a large degree of data that is not reliable, not reproducible, not consistent. At the same time, laboratory operations have really not changed in a long time. We still spend most of our time manually performing experiments, standing in the labs for hours, performing kind of small, busy liquid handling operations, for example, into a 96-well PCR plate. And at the same time, what this means is that we waste our most valuable resource, our scientists' time. The scientists should be able to think, hypothesize, read, understand, discuss results, and not be in the lab for 8 hours in a row performing cell culture. So looking at this from a different perspective, on the next slide, we can see how we think that what has really been happening over the past few years is that we have a divergence between the pace of progress in all other industries, where new technologies have really changed these industries with electronics, computers, mobile, 3D printing, machine-to-machine communication. And on the other hand, within lab research, we still have more or less the same work mode, more or less the same approach that we had 50 years ago. So if we look at a lab from the 1960s and a lab today, they look quite similar. Kind of you have a lab bench and you have kind of people performing the experiments by hand. So of course we now use hopefully electronic lab notebooks, and there's a PCR machine, and there are kind of 2 or 3 new pieces of kit. But the work mode itself has really not changed. And on the next slide, we see how we think about these things very differently, where we say scientists should be enabled to focus their time and their attention on higher value tasks, thinking through the science, thinking through their experiments, and leaving experimental execution to machines. So we've built a robotic platform where we use automation to generate high quality, very deep, very rich data sets within cell biology, within molecular biology, within biochemistry, so that we can enable scientists to spend their time with these higher value tasks. So our core beliefs, basically summarized on the right-hand side, is that in the end, drug discovery is always a series of decisions which have to happen ideally within condensed timeframes, and that only robust and reliable data can power the right decisions at the right time. And our answer is end-to-end automation. So it's very important to bear in mind that this is not high-throughput screening. We know high-throughput screening, everybody kind of has it, everybody uses it, 1 million molecules screened against a single assay. This is about end-to-end automation of entire drug discovery functions. So for example, running a target validation project entirely automated. siRNA, CRISPR, Western blotting, PCR, kind of all these different techniques being run in a consistent, reliable, robust fashion. On the next slide, we can get kind of into the details kind of a little bit more, where we see that Ulysses, the name of our core technology, indeed brings together biological expertise, biological experimentation with automation and robotics. And as I mentioned, we primarily work within biochemistry, molecular biology, cellular biology, and we support drug discovery from target validation to hit finding, hit to leads, lead optimization, etc. So the whole journey is being captured with our core technology and with the platform we've built in Oxford. In terms of working model, we primarily work with biotech and pharma companies, for the most part in North America, Europe, Asia Pacific, which is where we're mostly active, where our partners come to us and we then design an entire drug discovery project with them. For example, target validation, for example, a to lead campaign, and we then design all of the experimentation. We encode the experiments on the robotic system and then run all of these experiments in Oxford with superior quality, superior efficiency, and superior outcomes. And that is something which only automation enables us to do. So the core reason why we run experiments this way is because we want to make sure that experiments run on a Monday and on a Tuesday show the same result. And right now, that's simply not the case. So it really is about generating higher quality data and using these higher quality data to make better decisions. That is basically the entire philosophy behind what we're doing and what we've built with Ulysses. On the next slide, we can just have a very brief look as to what it actually looks like on a single experiment level. And that is just a very simple illustration. On the left-hand side, we see a traditional biochemical IC50 assay, something all of us are familiar with, 10, maybe 11 dose points of a novel molecule, looking at how robust kind of inhibition is in terms of percentage. Again, 10, 11 dose points and with very little information, especially once in isolation, this assay doesn't really give us much value. On the right-hand side is our approach to the same assay, where we generate more than 100 times more data points. We have far more dose points we can test because we can practically print every single concentration with our technologies. And at the same time, we can also capture what we refer to as experimental metadata. For example, equipment status, exact timings, humidity, temperature. In other words, there is a whole package of data that surrounds an experiment, which as of now is not being captured, where we say it is extremely important to know that, for example, overnight incubation was 7 hours, not 14 hours. Right now, there's far too much ambiguity in experimental data, and that is something where automation, including automated data capture, data analysis, and data processing, is really a quantum leap when it comes to how we think about data and drug discovery. On the next slide, I'm just going to briefly say who is behind the company. So it's a team of scientists, engineers, life science executives, primarily based in Oxford, but as I mentioned before, also very active in North America, very active in Asia Pacific. And we're extremely proud of having brought together a team that has worked in large pharma for 10, 15, 20, 25 years, and paired that with an incredibly experienced team on the robotics and engineering side. And next slide, to summarize what we think really is the future of drug discovery, the future of data generation, I just want to very briefly summarize that we firmly believe in expert guidance, in streamlining experimental design in a way that there's a combination between people who really know exactly what they're doing on the biotech and pharma side, coupled with technology. Because we believe technology is key to running processes in a more efficient way, in running projects in a streamlined fashion. And that is where the robotics comes in, reducing cycle times and making sure that experiments are experimentally consistent from run to run. So just to give one number here, we've run biochemical experiments from from compound received to full report generated, all data quality controlled and reported back within 24 hours. And that's something that is simply impossible in a manual setting. And so finally, tying it all together is having all of this data stored and accessible in one singular form. So instead of having to deal with diverging data formats, everything is collected in one place, accessed in one place by a secure portal. So our partners in biotech and pharma know exactly where the data is, they can use it, they can share it with their internal teams, and everything is, again, collected, stored, accessed in one singular form, including all of these very rich and important metadata. So that's the final slide. That's basically just the summary. And I hope you find it interesting to learn a bit more about what we're doing at Arctoris.
Thanks, Martin. That's really great. And I think that really ties together exactly what all the three of your technologies represent. So we can see that the concept of reproducibility, of understanding the science that you're looking at, but also being confident that it is good enough and controlled enough that you can then feed it into the computational systems to understand even further. The quality of the data is always going to be paramount if we're going to rely on it in order to build those sorts of models that Ajay was showing us, or to provide those sorts of manipulations that Kam has also detailed. So I think perhaps then, kick off the Q&A, we could look at perhaps, Kam, is there a question that you would specifically like to highlight to Ajay and Martin to say perhaps how you feel that their technologies can interact with your own?
I mean, for me, you know, I mean, I think if I go back, I think history is always a great teacher. And, you know, I've kind of probably come to drug discovery in the early 90s and, you know, all the learnings from the past. I think Ajay's point about, you know, looking at the mechanism of action, understanding using computational tools. I mean, Martin, in terms of data consistency and, you know, using robotics, allowing more thinking time for scientists as well. But really for me, it always comes down to the biology. It comes down to what, you know, fundamentally we go back, we've used overexpression systems, biochemical assays in the past, CHO, HEC293 with purified targets expressed. So for me, the closer we are to human biology and translatability, that is a key component of the whole drug discovery process. And I think technologies like what Martin has alluded to and how you can bring that downstream, and obviously with what Ajay's demonstrated about looking at mechanism of action, I think all these things tie together with other technologies which are very, you know, linked to machine learning and AI. I think AI and machine learning is here to stay. I think it's how it's utilized within drug discovery and at different points, junction points, is really the key. And what we derive and the output. And what is output? It's better drugs, quicker drugs, more relevant drugs, but fail less in the clinic. That's it. That's what I would sort of gauge from what Martin and Ajay.
That's excellent. And I think it all ties together. And I think ensuring that those connections that you talk about there between companies that are handling the information as well. So in fact, we have a question in from the audience, Martin. So how do you handle data storage and data access to ensure that's ethical, and the ethical consents, any ethical consents that are required and connected to the data? Although that's come in for Martin, obviously that applies to all three.
Yeah, I mean, first of all, I should add that we do not work with patient materials. These are not patient data. Instead, these are data generated from cells on the protein level. These are biochemical data, etc. So in other words, ethics approval is something which does not really apply in the space we're in. However, what is extremely important is that all the data is being generated is stored in a database, basically similar kind of to a LIMS system, which can then be accessed by the respective biotech or pharma partner. So in other words, they have a secure login, which gives them direct access onto the data. So they can see them, download them, analyze them, etc., etc. So that's basically the way kind of it works, basically via a secure kind of data portal, which gives direct access to the data. But again, ethics approval in our case does not play a significant role.
Okay, that's great. Hopefully the person in the audience will be happy with that. So again, going back to thinking about the connections that Kam highlighted there, Ajay, I can see that would also be important for your work. You're relying a lot on historical data and building new data into your models in order to put them into your systems. So how would you go about ensuring that the data coming in is accurate, is reproducible, and can be used to the best way possible?
Yeah, and I mean, there's always challenges in reproducibility, and that's why I think Martin's systems are very exciting, because it addresses that. So I mean, I guess what we do is, you know, we've got to make sure it's repeatable. So everything we do is done several times to the highest standard we can do. So yeah, we never just go by one set of experimental data for any of our modeling work we do. So yeah, we like all good scientists, we recognize that reproducibility is a key when it comes to looking at processes in cells.
Yes, and hopefully, I think the key to future drug discovery is being able to get that reproducibility in an efficient manner, which I think both Martin and Kam are certainly progressing towards. And I think that obviously the models that you're building, Ajay, will help to provide that, as well as requiring it going forwards. So, Martin, would you like to make any comment on how you feel that your technology can advance the future of drug discovery?
Very good point. So I think there's two things in here. So one is that all of drug discovery comes down to making go, no-go decisions. Taking this molecule forward, taking that molecule forward, taking a third molecule forward. It's always the most important decision is which program to progress and which program to stop. In other words, it's all about decision points. And for those decision points, we need robust, reliable, reproducible data. Because otherwise, it's just kind of as we could just kind of flip a coin if we didn't have kind of solid, rock solid data to stand on. So that's something which is true for all of drug discovery, traditional biotech, traditional pharma. But at the same time, as Ajay has also been referring to, there's the rise in AI-driven drug discovery. And that is a space which is seeing a lot of activity, a lot of growth. And within AI-driven drug discovery, it is even more important to train models on robust, fully annotated and structured data sets. Because within the machine learning community, there's this famous mantra, garbage in, garbage out. Because a machine cannot tell apart good data and bad data, the machine will use whichever data you feed it. So it's extremely important in order to get good outcomes, good predictions, good new targets, or basically new molecules, we need excellent input data. And that's where we see a very interesting synergy between robotics automation on the one side and AI machine learning on the other, which is exemplified by kind of many partnerships we see, which is exemplified by companies like Recursion doing a massive IPO, because they're really tying in the robotics automation and the AI ML. And that's something which we see is going to be an extremely important trend in the next 5, 10, 15 years.
So that's really interesting. Thank you, Martin. So we have a question in from the audience specifically for Kam about the breadth of your technology and the details of different types of cells that you're able to generate and validate with the IPSC with OptiOx technology.
Mm-hmm. Yeah, I mean, right now, the company was founded by Mark Cotter, who's a spinal surgeon, but also a neuroscientist. So, CNS is really, CNS cell types are really at the heart of what we're doing, but we're doing so many other cell types, immune cell types. We're working also in hepatocytes as well. We're working across different key cell types, which are whether they be involved in the metabolic disease, whether they be involved in cardiovascular disease. So yes, our technology has application across multiple cell types, but it's not just the parental, it's also the disease mutation as well. And it's working with partners in co-development partnerships to develop very sophisticated, not only single monoculture type models, but also co-culture systems. So as you know, organoids, spheroids, these 3D systems are much more representative of human biology than if you're just looking at a single, population of cells. So for us, more complex models, and also using technologies like 3D bioprinting as well. And for me, I mean, I absolutely echo what Martin has said, about reproducibility and how robotics and data driven, but also I think there's other emerging technologies that we have to embrace. And this is not just about CRISPR and iPSCs, but there's also digital pathologies and how translational medicine drug discovery can be used in a more holistic way, because they're looking at phenotypic assays. There's a resurgence on phenotype rather than just looking at single cells. So I think for us with bit.bio, we're looking at a much broader offering, and not just looking at individual cell types, but more complex, more disease relevant systems.
Okay, so there's a follow-up question from the same person in the audience. Would you be able to use your technology to reprogram cells for transplantation?
That's a very good question. Well, that is an area.
Breadth of uses as well as
that is a very. I mean, I'm not going to obviously bake any confidence here, but this is really, you can see from the heart of the platform. You know, we are making cells. Those cells can be used in a multitude of ways, and one of which is cell therapy. And yes, we will be announcing our first cell therapy partnership. I met with a major top 3 pharma company in the immune cell space, you know, which I'm not going to say anything more about. But the answer to this question is, yeah, I would love to. The answer is yes. We have our own programs. We're looking at in the regenerative medicine space, where of course our technology has applications in a cell therapy way. And so, as I said, this is, you know, we're a cell coding company. We're a cell coding company, but that cell coding works across basic research, preclinical, and cell therapy.
Fantastic, thank you. So perhaps we can ask the same question over to Ajay about the breadth of applicability. So you showed us the beautiful example of the E. coli in your model, and we would have seen more if the videos had been working as well, of course. But can that be applied across any cells, providing you have the information fed in an accurate manner?
Yeah, no, it's a shame you can see the video. So that like a video says 1,000 words, really. But one of the key points that I wanted to get across was that you know, by going down a computational modeling, a mechanistic modeling approach like we do, it's the things that don't go according to plan which are most interesting. So, for example, you know, I'll give you an example in the bacterial world that we worked on. So we looked at the LPS pathway in detail, modeled that, and according to our model, you know, it basically stated that this particular substrate should go up when you inhibit this particular enzyme, for example. And then when we actually did it in the lab, that didn't happen. So complete opposite happened. So that told us that there's some sort of new mechanism going on that's undiscovered. So what we could then do was really dig deep into that pathway, into that area, and figure out what was going on. And then it's an iterative process. So you're going round and round and figuring out why the predictions you get in the computer model doesn't reflect what's happening in the wet lab. And eventually you'll figure out the mechanisms that allow the predictions to be more accurate. So it's that type of process. So we found new regulatory mechanisms in the LPS pathway, which, as you know, have been studied for decades, and we've published on that as well. And the same with the MAP kinase as well. We're getting inklings on new regulatory pathways there as well. So I think this modelling is shining a lot of light in areas that that are hard to that have not been addressed before from a regulatory perspective. So yeah, I think it's a very the approach is a very useful approach, and it's actually delivering. Like I said, we've got early stage programs based on the targets that we've identified using the model. And I guess if we compare ourselves to other industries like the aeronautical industry, for example, people design, aeronautical engineers design thousands of planes on a computer before they actually build one, and that's what we're trying to do in with our computational modeling. We carry out thousands of simulations before we actually go out and test that in the lab, and then eventually in humans. So that's where we see our approach, basically, in terms of modeling.
It's proper basic science. You form a hypothesis first, and then you can go out and test it.
Exactly. Yeah.
No, that's really good. And I think that's, again, that's something that links everyone together, because once you've tested it, of course, the Arctoris systems can test it in a very reproducible manner. Kam systems can provide that extra bit of programming to then feed into your work. And I think it's a good overall image of where the future of drug discovery can lead. So I think that that's a good place to start wrapping up. I'd like to thank everyone for taking part. So do you have any final words? If you'd like to give a final sentence each, and we'll go in the order that we started in, Ajay and then Kam and then Martin, for where you see drug discovery in less than 30 seconds each over the next 10 years. Ajay?
Yeah, well, I think that, like I said right at the beginning, I think we've got a lot of biology to still discover. So, getting a handle on the biology is key to drug discovery, and being able to do that through computational methods like us, what we do, and through using techniques that Martin has, and exploring biology the way that the Kam's group do as well. I think that is the key to drug discovery.
Perfect. Kam?
For me, I think there's clearly a significant role for AI-assisted analytics and their workflows, the impact that they can have across the whole of drug discovery, from target discovery using technologies like CRISPR for novel target ID validation. I think more relevant disease models based upon human biology, which are more translatable. And I like to that new digital pathologies as well. I think it's an exciting part. I mean, as I said, I lived through the 90s. I think we're right at the cusp of something special now. And if, bringing together all these technologies and where they can impact, I think it's an exciting time.
Thank you. Martin?
I'd like to fully kind of echo kind of the last point that Kam made. I think what we're seeing right now is really convergence of different trends coming together. And yes, I think the most important one is biology meets technology. That can be robotics, that can be AI, it can be kind of many other things as well. But I think right now is really this time of convergence between these different trends. And so it is indeed a very exciting time ahead of us.
Okay, that's great. So it really is about connections, about understanding, and about building on what we already have. So thank you very much for all of you taking part. And I think that wraps it up nicely. So thank you.
Okay, take care.
Thanks.
Well, thank you, everyone. That was a really good session, good presentations, and sorry, Ajay, for the videos, but I'm sure we can make them available for the ones who are asking, because I saw them, they're great, and really good wrap up on how you're seeing this future of drug discovery. I made some notes too, and I had convergence between biology, engineering, technology, and other, which is bringing to the Kam's point about scaling, and scale brings needs for data and handling this data. And so that brings to AI. And final point was about aeronautic. Why? I think it's just to highlight, thanks for the analogy, AJ, but that it's good also to get inspiration from other sectors that are successful in that space. So thanks a lot. We're going for a break now, so you can grab some food if you're hungry, or you can also keep screening the app for making new connections, looking at the innovation workshops happening towards the end of the week if you want to sign up for these. And of course, we are coming back for the live stream in half an hour at 1 o'clock. Thank you very much.